Next, we have another former UCSD postdoc, currently faculty member at Johns Hopkins University. This is Kavita Rangan. It's again a pleasure to introduce her to this event. Well, she's going to talk about something interesting in cephalopods. They do a lot of RNA editing, and she's going to talk about that. I guess she was kind of fated to do RNA editing. There's an edited RNA in her name. It's RANGAN. So so let's hear about RNA editing in cephalopods.
Thanks. Thank you so much for inviting me to talk. Like Alok said, I just started a lab. One focus on the lab is on this RNA recoding, which is a pretty cool strategy that some organisms like this longfin intro squid use to generate basically protein variation in a context-dependent manner. And so we're sort of interested in how they use this recoding to adapt in response to changing environments. And so since I sort of just started, I'll tell you about some of my postdoctoral work that sort of lays the foundation for some of the directions that we're now taking in the lab. So starting here, which is just to say that, of course, a single genome produces, a myriad of different phenotypes, and the central dogma provides us with sort of a molecular framework for understanding this. And of course, the regulation of this process produces phenotypic diversity as well as phenotypic plasticity. And one form of regulation that's found across all animals is RNA editing. And so this is where enzymes called ADARs, I won't use this anymore, I guess, convert adenosine to inosine in double-stranded RNAs. And when this happens in coding regions, the inosine is read by the translational machinery as a guanosine. And so this can result in non-synonymous codon changes, which for the rest of this talk I'll call recoding. So this basically means that editing can specify protein variants with different amino acid compositions and different functions. And so, we're interested in RNA recoding in cephalopods, squid, cuttlefish, and octopus, which, are obviously charismatic animals with large brains and highly complex behaviors. And the reason why is because a body of work from Josh Rosenthal and Ellie Eisenberg's labs have shown that cephalopods use A to I editing to generate non-synonymous codon changes across their transcriptomes. And so while in humans and other common model organisms, editing occurs mostly in non-coding regions, in cephalopods you find close to 2 orders of magnitude higher levels of recoding. So near 100,000 recoding sites across 60% of ORFs. And so, two just very big questions that come out of this are sort of like why and how. So, what are the functional consequences of this high-level recoding, and sort of what are the mechanisms that regulate it? And so one idea has been that all this recoding really allows cephalopods to dynamically regulate protein function in different conditions. And so, so today I'll just tell you about some of my work that gets at this, which is just showing that how squid use recoding to alter protein function sort of quite acutely in response to their environment. And so going into this, I first wanted to see, what proteins and processes are highly edited in these animals. And so here I'm just showing Go terms that are enriched amongst edited proteins in squid. And then you can just see that terms related to sort of transport and localization are overrepresented, which might reflect that editing is involved in regulating these processes. And so I turned to microtubule-based motor proteins as a quantitative model to really explore the effects of recoding on protein function. And I worked with Sam Rick-Peterson's lab, worked in Sam Rick-Peterson's lab at UCSD, and they really specialize in motor proteins and mechanisms of intracellular transport. And so just to briefly say, dynein and kinesin motor proteins are very large protein complexes that transport diverse cargoes in the cell along microtubule tracts. And these are really highly conserved proteins, and we know a lot about them both structurally and mechanistically. And these complexes are very heavily recoded across cephalopods. And so here I'm just showing the number of recoded sites that have been identified by RNA-seq across multiple cephalopod species across these complexes, and just to say that there are hundreds of sites. And so I was interested in basically how recoding might be involved in regulating motor protein function. And so For this talk, I'll just focus on my work characterizing recoding of the kinesin 1 motor domain in Dorytoothus squid, which I used as a model. And just to say, Dorytoothus palei is the species where kinesin was discovered and isolated by Ron Vale and colleagues. And so this early video on top shows beads bound to kinesin from squid cytoplasmic extracts walking along a microtubule. And then below that, just to show that today we have a much more detailed molecular understanding of kinesin motility, like this animation from Janet Owasa beautifully shows. And so, one thing I was interested in is basically exploring if recoding is being dynamically employed in different conditions. And one environmental factor that immediately comes to mind is of course, temperature. And so temperature has been shown to globally affect editing in other animals, and so this has been seen in flies, in hibernating squirrels, and more recently in zebrafish. But how this affects sort of the specific protein function isn't, in these animals, isn't clear. And in cephalopods, editing levels had been shown to basically correlate with temperature across different octopus species, but it wasn't really known if recoding was acutely responsive to temperature and how that might be used. And so I focused on recoding in Dorytoothus opalescens, and so these are California market squid. And these squid are basically found in coastal waters along the Pacific from Baja to Alaska. And they spawn off the coast of San Diego, and so that provided me access to animals. And temperature is a very relevant sort of ecological factor for this species, so they migrate in depth every day, and so this data here is from Monterey Bay showing remote submarine sightings of market squid over the years, and the blue line is basically showing water temperature, and so you can find these animals in very cold water, down to 6 degrees C. And so from sequencing, I identified 14 edit sites along the kinesin motor domain for this species, and so the question really was, does recoding of the kinesin motor domain sort of change with ocean temperature, and how does that affect function? And so I collected wild squid egg casings, and after animals hatched, transferred them to tanks of different temperatures, and then looked at kinesin editing levels. And what I found is that editing at most sites significantly increases in the cold. And so every point here is an animal exposed to either 6 degrees in blue or 20 degrees C water, and then along the X are different recoded sites along the motor domain. And so this change in editing that we see is within 1 day at the given temperature, showing that recoding in squid is really acutely sensitive to temperature. And I've since done more detailed time courses and find that the changes in editing can occur within 1 to 6 hours depending on the transcript. And so, so to understand kind of the effects of editing on kinesin function, I wanted to know, what kinesin variants are really being made in animals. So in other words, how do these sites actually exist in combination along individual transcripts? And then are there different variants made at different temperatures? And so one way to sequence individual transcripts with sort of high accuracy is to just clone out cDNA from squid and sequence hundreds of clones. And so that's what I'm showing here, which is just every row here is just a different clone sequence, and the column is basically colored if that site was recoded in that sequence. And so these are basically representative of individual transcripts. And we can see that recoding is really producing many different combinations of these sites in individual transcripts. And so I did this in animals exposed to either 6 degrees C or 20 degrees C water. And so this is just a summary where you can see the percent of transcripts that have increasing numbers of edits in them. And so just to say that you have a much higher percentage of fully unedited transcripts at 20 C. And then there are more multiply recoded transcripts in animals exposed to 6 C. And then here I'm just showing the unique variants from each temperature. And so you can see that recoding is really producing many different temperature-specific variants. Okay, and so, we're interested in how do these variants basically have altered kinesin activity. And so here I'll just focus on one highly edited cold-specific variant and compare that to unedited squid kinesin. And so to evaluate this, I measured kinesin activity in single molecule motility assays where polymerized microtubules are adhered to a cover slip and then fluorescently labeled motors are flowed in. And then we can monitor the movement of the motors using turf microscopy. So this video is showing GFP labeled kinesin walking along a microtubule. And then from a video like this, we can make a chymograph where you can see the movement of the fluorescent motor over time, and then we can analyze these chemographs to extract different parameters of motility. And so I did these assays at 25C versus 8C using a Peltier temperature controlled stage with like a fitted copper-plated slide cap, and then directly measured the temperature of the slide close to the imaging point with a thermocouple probe. And so if we look at the activity of human kinesin at these temperatures, we see sort of as expected that the activity of human kinesin is highly temperature sensitive. And so on the left, you can see that shift from 25 to 8C for shorter run distances, and then on the right are histograms of velocities, and you can see slower velocities at cold temperatures. And I should just point out that sort of the magnitudes of these effects are sort of similar to strongly defective motor mutants. And so now if we look at unedited squid kinesin at 25C versus 8C, what we see is very similar to human kinesin. So you see significantly slower velocities, shorter run distances, and then on the far right, you can see landing rates, which is basically how often kinesin is binding to the microtubule. So you see much lower landing rates in the cold. And now if we look at this kinesin variant that's made exclusively in animals exposed to cold water, you can see right away from the chymograph at the top that it has significantly longer run distances and much higher landing rates compared to unedited kinesin in the cold. And so that's quantified below. And I think this is interesting because it shows that recoding in response to cold temperature is really specifying a motor variant that's able to bind microtubules better and travel farther in the cold before falling off. So basically kind of going from a non-functional motor in this context to a functional motor. And I looked at the motility of other cold-specific variants and saw an increase in landing rate in those as well. And so, I think overall this kind of shows that editing in squid is sensitive to environmental temperature. It specifies protein variants that may support function in different environmental conditions. So now, if we look at this cold-specific squid variant, you see it has many different amino acid substitutions. And so I wanted to basically ask if those effects I was seeing in squid kinesin were conserved. And so I focused on individual sites that were both preferred in the cold in squid and also in amino acid residues present in human kinesin, and tested those individually now in the context of human kinesin. And so this is showing wild-type human kinesin, which, like I showed you earlier, in the cold has shorter run distances and also lower landing rates. And then if we look at these individual substitutions that are cold preferred, you can see that each of these substitutions alone has the effect of increasing run distance and landing rate of human kinesin in the cold. And so I think this is interesting because it means that basically these edit sites from cephalopods can show us really specific amino acid substitutions with conserved functions in human proteins. And I think one surprising thing here is that even in a protein as well characterized as kinesin, these squid recoding sites are really pointing to very specific modulatory substitutions that we wouldn't easily predict from structure and that haven't been characterized before. And so here I've just highlighted those sites from cold edited squid kinesin that have these effects on the structure of human kinesin. And so one of these sites, the one tyrosine 77, is in sort of a critical region known to be important for motility, while the other two sites are in sort of less well-characterized areas. And so just to sort of briefly say, kinesin is a dimer that moves along microtubules in this hand-over-hand mechanism, and ATP binding of one head leads to docking of this flexible neck linker onto the motor head, which sort of pulls the trailing head forward along the microtubule. ATP hydrolysis releases the head from the microtubule, and that cycle can happen again. And this process is really dynamic, and so bias diffusion and sort of the docked and the undocked neck linker states of ADP-bound kinesin are important for the sort of directional processive movement. And this tyrosine 77 is highly edited across several species of cephalopods, and it lies exactly where the neck linker docks onto the motor head, which is highlighted in orange in the structure. And this docking is really critical for motility, and disease mutations decrease activity. But this tyrosine to cysteine substitution increases run distance and landing rate while maintaining velocity, showing that sort of editing may be able to sort of fine-tune this motor property in some way. And these other two residues that we find is important for cold function have been identified separately as likely important in sort of the dynamics of stepping. And so basically all to say that sort of the substitutions that we've tested sort of experimentally validate the importance of these residues. And I think this illustrates an idea that our lab is now building on more broadly, which is that we can use recoding sites to explore protein function and find important sites of modulation by looking at sites that are highly conserved yet recoded in cephalopods and then ask how they affect function. And I think this is a particularly useful approach for proteins that are really large and complex, for which things like deep mutational scanning, for example, wouldn't be possible. And so we're applying this idea more broadly, which is just using recoding sites as kind of nature's guide to exploring the function of conserved proteins and processes. And we're trying to build this out as a resource by mapping cephalopod recoding sites from published RNA-seq data sets across species and conditions onto conserved proteins and predicted structures on a proteome-wide scale, and have this as sort of a searchable website, because I think this will allow us to really generate new hypotheses about protein function and interactions and molecular adaptability. And I'm out of time, so I'll just say that I think that this gives us the ability to kind of identify sites that co-vary across a protein process or pathway, and then it can also highlight structural interfaces both within and across proteins. And I won't go through some of this, but we've taken a look at some of this in the context of temperature-sensitive editing globally. And so, yeah, and so just as a brief sort of mention, we are going in a very different direction now in the lab, looking at how editing is used to recode DNA replication and repair machinery, and this is a really exciting direction that if you want to come talk to me later, I'd love to. And so, yeah, thank you. I'd like to thank Sam in the Rick Peterson lab, where I did all of the work that I showed you, as well as Josh Rosenthal at the MBL, which is, he's the reason that I first got interested in cephalopod editing, and to all of the funding that has supported this. So thanks.
Thanks, Kavita. Let's take some questions. I'm again going to try favoring early career researchers. I'm actually not sure if you're an early career researcher, but go ahead. Okay. Okay, fine. Yeah, we have, I'm sorry, yeah, name and question.
I'm Kira. I'm at Brandeis University. So a couple of questions. The cold-specific edits seem to work better than the unedited versions at cold temperatures. Did they perform similarly to the unedited version at the worm temperatures, or why would you not always use them?
Oh, so they actually are, it kind of scales, so they are also equally have higher landing rates. At 25C, they also have higher landing rates and more run distance. And so I think this is maybe like a question of like maybe more is not always better is one thing. We also don't really understand anything about the turnover at the protein level and how also heterogeneity in this context is really used. And so, for example, kinesin is in the simplest sense, kinesin is a dimer. And so how would a heterodimer behave? Or how would a mixed population behave? And so these are all things that I think we can address, sort of start to get at experimentally in these sort of single molecule setups, but yeah.
And just quickly, do you have any idea what triggers the change in editing to a specific variant or another at different temperatures?
Yeah, so the enzymes that do the editing, so I should say that the biology, like the unique biology of cephalopod ADARs versus other animals, like mammalian ADARs, for example, I think is something that is ripe for a lot of exploration and groups are doing, including Jenny. But yeah, like the question of, so I think one interesting thing that we're trying to pick apart is like these specific combinations. So, for example, we don't know yet whether those combinations, like whether editing is happening co-transcriptionally or post-transcriptionally for that specific transcript. But we're playing around now with like RNA folding to see whether the sites that are co-edited are actually just close to each other physically, for example, could be one explanation. But yeah, there are many other sort of transacting factors that might be involved in these.
Had a question there, and then there was one more question. Something else? Okay, cool.
I'm wondering whether there's something interesting about the behavioral biology of the squids that is being reflected here. And so I seem to remember, it's not something I work on, that changing colors has to do with motor proteins.
Oh, yeah, it's interesting. So yeah, like myosins are actually some of the most are very heavily edited. Like if you were to normalize for like protein size, let's say. And yeah, I haven't explored that at all. But yeah, that's an really interesting question. I mean, that also comes down to how that plays into. I can't imagine that on the time scale of actually affecting like camouflage, but maybe setting up like the differences in editing, setting up sort of the larger patterns, for example, on the mantle. I don't know. Yeah, I don't think that that's been explored at all. Yeah.
Have more questions? One question over there.
Hi, thanks. Great talk. I guess I'm wondering if there's other compensatory changes in the general biology, right? So if your editing goes up and they're migrating with depth and they're changing temperature during the day, do they preferentially make transcripts that would be negatively affected by too much editing at the time of the day that would be ideal, I guess, when they're in warm water?
Yeah, I mean, I think this kind of comes down to the same, like the idea of basically what are the timescales that are relevant for this. And I guess I would say that in my head at least, I'm imagining it more as like there being a sort of heterogeneous population at all times of edited and unedited variants, and that those are just are being utilized sort of in different environments. So like the cold preferred variant, but yeah, like what are sort of the mechanisms that would favor one variant over the other is unclear. Yeah.
Cool. Thanks, Kavita.